Hello, everyone. Today, we're going to be talking about how anthropologists use chemistry, specifically isotopic signatures, to uncover hidden details about fossils from the past. Isotopes are versions of the same element, but with slightly different numbers of neutrons. This happens naturally, and when we analyze them in fossils, they give us all kinds of information like past diets, environments, and movement patterns. Stable isotopes are isotopes that don't decay over time, which makes them really good for studying ancient materials like bones and teeth. Anthropologists use teeth and bones specifically because tooth enamel forms during childhood and doesn't change afterwards, so it preserves the teeth's chemical structure in early life. Bone chemistry, however, does change over time. It reflects the diet and environment of a person's life. And by comparing isotopic signatures in enamel and bone, anthropologists can piece together details like diet of the fossil and the environment it lived in. But before we get deeper into isotopes, let's first talk about the fossil context. Fossil context is everything about where and how a fossil is found, like the type of rock sediment it was buried in, the different materials that surround it, and the kind of environment it's in. These give anthropologists important clues of how the fossil lives, and without these context clues, the fossil would just be a bone. Isotopes expand on the fossil context further. By analyzing the chemical makeup of the fossil, specifically the isotopes, anthropologists gain a further understanding of the kinds of food the fossil ate, what it drank, did it stay in one environment or move to a different one, and whether the climate was hot or cold. Isotopes are versions of the same element. They have the same number of protons, which means they're the same element. What makes them different is that they have a different number of neutrons. This small difference is what makes isotopes useful for research. On the left here, you can see carbon-12 and carbon-13. Both are carbon atoms, but carbon-13 has one extra neutron. This leaves a signature in bones that scientists can measure. We'll focus on stable isotopes which don't decay over time, which means they are still preserved well in ancient fossils. And we're interested especially in isotope ratios, which is basically how much of one isotope compares to another. It's with this comparison that tells us all the information we previously talked about, like diet, climate, etc., etc. Carbon-13 levels tell us about diet, like whether someone ate more meat or vegetables, and carbon-18 tells us about water sources and climates the fossil lived in. Now, why tooth enamel and bone? Tooth enamel forms in early childhood and doesn't change after that. And it's like a snapshot chemically of an organism's early life. So whatever diet or water source a person had as a child, it gets locked into their enamel permanently. Bones, on the other hand, are constantly remolding. It breaks down, it rebuilds itself at a constant rate. This means that the chemical signals in the bone reflect what the fossil ate, drank, and experienced later in life. When looking at the isotopes from bone and teeth together, we can build a timeline and compare early life to later life to see if diets change over time or if the animal has moved to a different region. One of the most important things isotopes tell us is what the fossil ate. When we eat plants or eat animals that ate plants, the carbon from that food ends up in our bones and teeth. Scientists look at the ratio between two carbon isotopes, carbon-13 and carbon-12. Different types of plants absorb these isotopes in different ways. C3 plants like fruit nuts, berries, leave behind lower carbon-13 rates. These are common in wild environments, which is a typical hunter-gatherer diet. C4 plants, like corn, sugar, cane, leave higher carbon-13 values. These are mostly farm crops and show up when agriculture becomes part of somebody's diet. So when scientists study carbon isotopes in bones or teeth, they can figure out if someone relied more on wild plants or farm plants or both. This helps us understand how diets change over time. In addition to diet isotopes, can also tell us about the environment the fossil was in or may have moved to, and for that we use oxygen isotopes. Oxygen 18 and oxygen 16 are absorbed into our bodies through drinking water. Different water sources have different oxygen isotope ratios, depending on things like elevation and climate. The oxygen signature in somebody's bone or teeth reflect the region they live in or lived in. This map over here shows predicted oxygen isotope values around the world. This specific one is based on cotton plants, but the pattern is same for humans because it shows differing water sources. You can see that different areas like the coastal and inland regions have different oxygen levels. Tooth enamel locks in the oxygen signal from early life, while bones update it as a person ages. So if we see a change in oxygen isotope ratios between a person's teeth or bones, we can tell they moved during their lifetime. This is useful when it comes to tracing migrations of ancient humans and environmental changes as well. Just like all scientific methods, isotope analysis has its limitations. One big issue is diagenesis, and that's when chemical changes happen to a fossil after burial. Over time, things like groundwater can alter the isotopic signals in the bones, which makes it harder to interpret it accurately. There's also contamination. Poor handling in the lab or contamination can interfere with readings, so scientists have to be careful with how samples are collected and handled. Sometimes diets or regions can produce overlapping isotope signals. This makes it difficult to say for sure where someone lived or exactly they ate, or what exactly they ate. In cases like this, isotopes don't give a direct yes or no answer, but rather need to be interpreted alongside other evidence. Also, sample quality really matters. If a fossil is too degraded or poorly preserved, the isotope date might be damaged or ruined. And it's important to know isotopes don't tell the whole story. Rather than fact, they give anthropologists clues like different pieces of a puzzle. Anthropologists need to fill in the rest of the data with other kinds of data. So why does any of this matter? Why should we care about isotopic signatures in teeth and bones? Well, because they help us learn more about the past that we otherwise wouldn't know. Isotopes reveal what fossils ate, where they lived, and how they moved, even when we don't have any sort of records or artifacts. They also help fill in the gaps in the fossil record, where bones might be incomplete. Isotopes give us chemical clues that complete the story. Most importantly, isotopes connect individual lives to historical changes. Things like agriculture, migration, and climate shifts all show how people adapted to changing environments and how communities evolved. And that's the end. I hope you enjoyed my presentation. Thank you.